here we are. Okay, so yeah, apparently I should have moved this up, this picture up. Yeah, that's what that's the slide that I was missing. Okay, so one more time, same viewpoint. This time you can see bump on the left. Okay, and basically what happens is this is another interesting story. Okay, and it's due to Valery Lukyanov, who's a professor at the university, right, Lobachevsky University, and my dad, and he was Lukyanov was a student, and this is called the cell node periodic orbit with non-central homoclinics. Okay, so unlike the I always say that we shouldn't do that, right? But I'll do this. So unlike the blue sky catastrophe, right, when this two-dimensional unstable manifold of the cell node periodic orbit, this one, right, it comes back on the left from this called strongly stable manifold, right? So that's what makes the blue sky catastrophe unique, and one of dimension one, and secondly, right, it results in the emergence of the a single stable periodic orbit. Okay. In contrast, now we consider the case, what's called the boundary case, when this looks like a, okay, I mean, given that most of you are Russian speaking students, right, this looks like a hobbit of elephant. Okay, I'm pretty sure you don't know what hobbit means. I mean, you know that hobbit means this kind of elephant, and this time this two-dimensional surface, right, comes back to the cell node periodic orbit transversely to its what's called strongly stable manifold. Remember, this gray surface strongly stable manifold, it separates this cell portion of the cell node, and this is called nodal or stable portion of the cell node. If it happens to be on the left, you would converge to the cell node from the left, right? Otherwise, you will escape to the right, right? And then when you come back, what you can do, right? So clearly, this gray disk, right, is two-dimensional, and even though the hobbit or this pipe shrinks by the time it comes back, nevertheless, right, its diameter is not really zero, right? So which means these two surfaces have what's called transverse intersection. Okay, and when you come back, you turn around this green manifold, right? Come back, and then you have two options, right? If you come back, say strictly on the left, then you would go to another region, right, to converge the cell node. Otherwise, you're going to straight back to the right. So the way you can analyze this case is you pick a cross section. Cross section is a little plane transfers to this manifold, I mean to the periodic orbit manifold, and in this cross section, the cell node is going to look like a dot. Okay, then say the upper intersection point, right, is going to look like this line, and that line comes back to the cell node along this what's called strongly stable manifold, and this is known as a homoclinic wiggles. Okay, homoclinic wiggles. But in the cell node case, clearly you're always going to go on the right. But if I lose the cell node right, then I'm going to see something like this, right? It comes back over and over. But the point is that when you decouple the cell node bifurcation into two, so one point on the left becomes stable, point on the right becomes unstable. Then depending on the on how you come back to the settle periodic point, right, or settle fixed point. So if you converge like this, right, always on the right hand side from the incoming manifold, then you're gonna get, by stability, you're gonna get this part stable, right, and then bursting part, right. If you happens to be always on the left, then you would end up on a stable fixed point, right, like this, right. But then there is always an intermediate moment that was depicted here and there. When you come back, you can go to the right, to the right, to the right, but then all of a sudden you're going to end up on a stable periodic orbit. So this picture on the left basically illustrates what you will see in the Hinman-Russian model, right, at the given parameter values. So you pick initial conditions in the bursting region, and then, okay, depicted here is just the very last 3 bursts. You never know how many bursts you will encounter first, right? It could be 1, 2, 3, or maybe 55, no clue. That's what makes it unpredictable. It depends on initial conditions. But eventually it's going to end up on a green limit cycle, because again, you're going to be on, happens to be, right, in this case when you either go to the right, but then eventually you're going to end up on the left, right? But this number of bursts transitioning towards the stable tonic spiking attractor is unknown, okay? That's what makes this situation very, very interesting, right? And also, when the saddle node disappears, then it's replaced by what's called hyperbolic subset, right, that has countably many orbits nearby. Right? That's what makes this bifurcation very interesting from the point of view of mathematics. Okay, so yeah, I was missing that slide. I'm glad I found it. So I guess misplaced it. Now this is the chain model. Okay, by the way, just to tell you what's called beta cells. Beta cells in the human body, they're responsible for what's called insulin secretion. Insulin, okay, for diabetics, it's a crucial thing, right? So you want to make sure that these cells, they generate enough insulin, or they command other cells to produce insulin enough for the blood. So that means the sugar level stays at the normal level, right? Not too high, not too low. So that's basically the model, what's called a beta cell. So you know what beta cell stands for. So speaking of cells and neurons, the picture on the left, what you can see here, is okay. This is called 2 neurons, right? Again, coupled. They call interneurons. So these 2 interneurons, dark blue and light blue, okay, you can see dendrites and axons, right? And basically they're coupled. They form what's called inhibitory coupling between 2 cells, right? And they're coupled by inhibitory synapses to produce what's called half-center oscillator. What is shown here is the recordings from somas, right? On electrodes poked into these areas, right, called somas. Okay, and then when everything is fine, right, when these interneurons, part of the what's called CPG, central pathway generator, that controls the locomotion in not locomotion, blood circulation in the leeches. Let me just see if I can show you some slides. So let me click Finder over here. Oh, let me see. Okay, so I go to Dropbox, then I go to Talks, right? And let me see. I'll show you a couple of slides just to Riva movie. Okay, so let me see this one. I mentioned to you that, okay, so let me see. You'll see a leech in a second, right? So that's a movie, that's a leech, all right? And it has nothing to do with the blood circulation, but it has to do with leech locomotion, how it moves, right? And medical leeches, not all of them, right? They have 2 types of locomotion. They can crawl, palsti, right? So when they crawl, they have a sucker, 2 suckers over here. So it's going to bend like this, right? And then there's a rear sucker, and it fixes it to the surface, right? And pushes the front sucker, so it bends like an arch, right? And then the sucker gets to the surface, right? And then drags the rear sucker, but the second part of the body towards the front sucker. That's why it's called crawling. It crawls. Okay, again, that's a style of swimming. It's called crawl. It means, I forgot already what I said. Crawl means palsti. Okay, and clearly, if you have a leech and leech wants to move from this point to that point, then, and there is no, and on the dry surface, it's going to crawl like this, right? Like this. The trick is that medical leeches are also swimmers. Not every leech can crawl, but not every leech can swim. When I say medical leeches, again, these are special kind of leeches that no longer used in medicine, right, or rarely, but they were used to by the French doctors during the Napoleon mission, when Napoleon went to Egypt, right, and then to Italy and then to Russia as well. So on the Russian mission, right, they had 500,000 leeches in the barrels. At that time, leeches were used to remedy headaches, blood pressure, and pain. Okay, so and then if you place a water, right, in that aquarium, what's called aquarium or pool, that leash is going to swim not like a snake. It's going to, like in a zigzag manner. It's going to zigzag, but again, it's going to zigzag not in a horizontal plane, but in the vertical plane. Okay, it's going to swim. Guess what? What if you have a level of water that is not deep enough for the leash to swim, right? But it's deep enough for the leash to crawl. So which is called the bifurcation level of water. So again, when the surface is dry, the leash is going to crawl. When the level is high enough, it's going to swim like this. So it's covered by zigzagging, right? And then when the level is intermediate, it will try to perform either one, right? Or you will see what's going to happen. So leash has a sensory neuron on the surface, on the body, basically on the back. And if the pressure that water puts on a sensory neurons is noticeable, which means that the level of water is high, then it's going to swim. But if the sensory neurons on the back of the leech don't feel any pressure, then it will give a signal to the CPG that controls the locomotion. Okay, now you got to crawl. And the point is that the question is that is it the same neural circuit that controls both locomotions, or if you want to swim, you use one circuit. If you want to crawl, then you have to redirect the input into the crawling CPG, right? And it's still unclear, but it's known that some neurons, they are active during crawling and swimming, which basically means that they're part of both CPGs, right? Or one CPG. So it's still, again, it's called a hypothesis, but they're active, and that's why people who Bill Kristen, he was director of neuroscience program in the University of San Diego, UCSD. Then he, I have a meeting in 15 minutes. Okay, forget about the meeting. All right. Okay, so, all right, let's see what's going to happen. Okay, you see that leash, it wants to swim, and then it crawls, and then swims again, and then crawls, and then swims again. So basically, you can see it switches back and forth between swimming and crawling, okay? Because it does know what to do, okay? This makes the experiment so amazing, right? So this is bifurcation level of the some bifurcation level of the water in a leech, in a pool. I'm sorry, right? Okay, this one. Now the other movie has to do with what we discussed earlier. Okay, so this is the leech, right? Looks like a dragon, and it's just a section of a leech, right? I mean, and what you're gonna see, this is nervous system here, okay? And leech has a heart, which basically it's a closed pipe like this, right? And the blood circulates in that pipe. And what you see here in a second, right? It's circulating blood. So you see it moves like this over here. Then it's going to re-emerge on the top, going clockwise, clockwise one more time, clockwise. You see it moves like this. And then it reverses the direction. Now it moves counterclockwise. Okay, and this happens every time, right? Regardless of the size of the leech. So the blood moves again 3 times clockwise and 3 times counterclockwise. Never like 2 and 3, 4 and 2, right? But always 3 and 3, right? Okay, and that's what that's why. Okay, and the reason why leech is well-studied animal, right? It used in different experiments, and this time, right now, we're going to focus on neurons that are part of the central pattern generator that controls the blood circulation. Okay, so but basically, what I'm going to show myself in no, basically what half center does. Imagine that the the heart of the leech, right, that pipe is an elastic pipe, right, made of plastic, right, and then when you push, if you want to push the blood, say leftwards, right, you have to press the portions of that elastic pipe, say leftwards or rightwards. That's what half center does. Okay, it pushes this neurons, right, or this sequence of neurons. If you want to make the blood circulate leftwards, right, you have to create this kind of traveling wave moving leftwards, right, by pressing this. Every episode of spikes makes the it goes to amplify neurons, what's called motor neurons, and then motor neurons basically compress the heart in a sequence that would end up in a clockwise or counterclockwise blood circulation. Okay, so, and the interesting thing is that the very first and original, known as a canonical model of a heart interneuron, right, it has 14 differential equations, and the people who put together the model, they didn't have any knowledge in the dynamical system, and mostly in mathematics. So they use Hodgkin-Huxley experiments, type of experiments, to find the constant, all these equations, right, and all they put together, and it becomes 14-dimensional, right, and then you can reduce it to dimension 3. So this is what's called minimal. It's a minimal requirement for the Burster model, and this is done not mathematically, but in a biophysical way. Especially, you apply particular drugs that block certain channels, and by blocking these channels, right, you decrease the number of equations in a model. So in the simplest model, that's how it's going to look like. Okay, so now we show, of course, right, we show pictures nowadays rather than equations, but I'll show equations. So you have C is a capacitor, is like constant 2, and then you have how many currents in the right hand side. You have a leak current, like integrating fire. Leak current basically is going to give you the steady state. Then you have what's called potassium current here, this one, and potassium current M. It's a probability. It has clearly M. It has M squared here, which is this dynamic variable that goes into this equation, which controls the current. Again, remember I said everything is minus. Maximal conductance, 2 gates, so probabilities are independent, and the voltage minus the reversal potential for the potassium current. Okay, then fast sodium. So again, you have H, dynamical variable, right? It quickly activates. So look at these crazy numbers. You have exponential function that has 500, but don't forget that voltage is in millivolts, which means 10 to the negative 3, and that 10 to the negative 3 compensates for this 500. So this exponential function becomes normal, right? So you quickly open up the sodium channel, right? And then this is called inactivation, which is again F infinity function, which is basically 0 and 1, and that's cubed because you have 3 channels, 3 gates for the channel. Okay, and it's a fast variable, and here potassium is slow. And what we need here, okay, the way you read it, right? Okay, so to be slow, tau should be large enough, which can translate into small epsilon. So when this F infinity function is 0, then M prime is dictated by negative M, which is basically exponential convergence to the 0 steady state. So M is probability between 0 and 1. And then F opens, right, at a certain level, and then this function becomes 1, which means that this probability, this value is going to be 1, which means M variable will try to get to 1, okay? And this is, again, F infinity function, which is sigmoidal function. And what's interesting is that sigmoidal function, this sigmoidal function, it has an intermediate value when probability is 1/2, so whether the channel is semi-closed, semi-open, and this occurs at the level of negative 18 millivolts. That's how you interpret this number. And what is shown here, this parameter that we're going to use it, right? It's a very, very bifurcation parameter in the system, okay? Meaning that when biophysicist or neurophysiologist found that the potassium channel is open at the level of negative 18 millivolts, how did they determine that number? They made a number of experiments. How many? Say 10, right? And then they average 10 numbers and found negative 18. What we use as a bifurcation parameter, it's a deviation from that mean value, okay? And that's our parameter in the system. So, and then altogether, it allows us to treat this neuronal model as a slow fast subsystem. So we have voltage fast and another variable slow, right? And z here is it's not a vector, it's just it's a scalar, it's a single z. And it has the same, you see, a shaped, right? This time is a S-shaped. Is it S? Yeah, S-shaped. Okay, knee point, this two-dimensional surface that begins with the Andronov bifurcation, and then by the time that surface gets to the end point, it robs back. You see, it robs back, and then the outermost part of the surface is made of unstable limit cycles and terminates through the Haber-Klinic bifurcation, right here. Okay, now remember, as I said, F is sigmoidal curve, okay, and it doesn't contain, it contains only V, which makes it this yellow sigmoidal curve, which ranges from 0 to 1, like this, it bends like that. Okay. All right. Okay, again, this is just an illustration. Okay, I flipped the curves, and basically this sigmoidal curve, and this, again, the whole idea is that by changing this shift, you move by varying this parameter, you shift this low incline up and down, but this time it's a sigmoidal curve, so it's going to bend one more time, like here. Okay, and what happens is that when the sigmoidal curve has a tangency here, right, then it corresponds to cellular bifurcation, then the burst duration, okay, that burst duration, maybe this the burst or the interburst interval may become very long. But as you can see here, for some value, it looks like this bursting, and then you change the parameter and burst duration increases even further, and then it becomes infinitely long spike. Okay, and the question is how that happened, right? And how we can explain what's going on. Okay, one more time, the same equations, these crazy functions. You see 500, negative 150 voltage. Okay, so again, don't forget that voltage is done in millivolts. That's why 10 to the negative 3, like this number, compensate for this craziness. So this function really changed from 0 and 1. Okay, and they're relatively smooth. One more time, this is topology of this manifold. Okay, branch of the equilibrium states. Okay, this sigmoidal yellow line curve surface. I'm sorry, right, and then this manifold that robs back. And in this particular case, for some parameter value, that folding point is going to touch the knee point, which corresponds to the Haber-Klinic saddle node that we also discussed in the classroom. Okay, so, and then you have two parameters. Okay, this V shift that shifts the yellow surface up and down in a parameter space, and the second parameter is external current. And bottom line is this kind of bifurcation diagram. So above this line that corresponds to cellular bifurcation, you have quiescent state. Here you have bursting, and then you have spiking activity here. Okay, and the transitions between activity types are very interesting. So let's focus on this picture on the right. So we transition through this boundary, right? Okay, so shown here in blue and red are two trajectories uncoupled. Okay, and as you can see, they repetitively, cyclically generate this bursting activity, but they slow down right here, which means this is Andronov bifurcation. Okay, shown here is a number of spikes per burst, and when number increases, then this red means that you have like 100 spikes per burst, and when you're here, you have like 2 spikes or 3 spikes, and then you accumulate to get more spikes per burst. Whereas in this region right here, you have bistability. So depending on initial conditions, okay, two trajectories, they diverge in space to converge to a bursting trajectory, blue one, right, and the tonic spiking trajectory, which is shown in red. So clearly in between these a threshold. All right, and okay, now let's talk about the topology of the manifold. What is shown here on the left, it's called parameter continuation algorithm. So basically, this is a GIF file, so it's going to end up and start over from here. So basically, I'm shifting this yellow null line up and down. Let me show it to you. It's going to show on fixed, but indeed is that I shifted from upper position to lower position. Okay, so let's wait. You see from here, and then I have all these limit cycles, and all together they form the surface that robs back right now to generate that burst or fold, and then it's gonna glue to the either saddle equilibrium state or saddle node that corresponds to this turning point on a manifold. Okay, so that's how you use cleverly, right, or intelligently the parameter continuation software, and that's what you're going to get a picture like this. Remember, I have these limit cycles found numerically, and what I've done is each limit cycle here, I plug it into this law equation here, and then averaged over the period of the limit cycle, right? And for some parameter values that corresponds to that position of this low null line, okay, what do I have? I have a slow equation that has basically it's driven by this function on the right hand side. So no surprise, it has a tangency here, right? It's like a quadratic function, and because the surface robs back, which means this function is not really a function, because it's not one-to-one, right? But we don't consider the slow part, or we need it separately, right? But basically, what I'm going to do next, I'm going to slowly lower the position of this yellow sigmoidal surface, right? In projecting, it looks like a curve, and these 2 points, one stable. Again, remember, stable, unstable. Unstable corresponds to saddle limit cycle, right? They're going to merge together. Okay, let me show you next. Right, so they're gonna right here. They're gonna merge together, right, and then disappear, right now, and then I have only one big zero in this, 1 0 in the right hand side, which corresponds to a big limit cycle. Or, okay, if I move this up, right, actually allow this up, right, when this red point and that black point, they're gonna also get together and then disappear through the saddle node, and then I have the only one stable green point that corresponds to this tonic spike in activity. Okay, so that's basically averaging a Pendragon-Rodigan theory in action. So you use it to identify whether the surface is transit for the solutions, or you have stable or unstable limit cycles in a system, and you need it, okay, because you want to find this tangency, right, that I discussed, that corresponds to the blue sky catastrophe in a system. So as soon as this double limit cycle periodic orbit disappears, I can transition through it ghost, right, and then I generate a very long burst. And the shown here is that as you approach this transition, right, the burst duration goes above 80 seconds, which is a lot, because typically, right, the burst duration should be in 2-3 seconds range, right? So that's the example of the blue sky catastrophe in action. So that's how I know that I have either limit cycle here that corresponds to tonic spike in activity, and then I have bursting activity after that. Okay, but it's good, but it's only good to detect just a one bifurcation, like saddle node bifurcation, because again, saddle node bifurcation by itself is generic for systems on in dimension R1, right, in one-dimensional space. But if you want to study, say, period doubling bifurcations and other things, right, you can't. I mean, again, averaging has its own limitation. You have to move forward and find better tools, right? And whenever we deal with periodic activity or cycling activity, then return maps, the technique of Poincare return maps is, oh, Jesus Christ. My daughter is coming for the Christmas next week, and she said that don't buy blini for the caviar. If you know what I mean by that. Покупайте блины для икры. All right. Okay, so it's so funny. I'm sorry, guys. All right. Okay, the way you do it, once you detect this manifold, right? So you have all this, again, that surface, right? Then what you do is that you find on that manifold, you find this line that corresponds to voltage is minimized. I showed you before that, right? And then say it's made of 1,000 points, right? And then from every point you release and find where, after making a single turn around this surface manifold, where the point lands on that space curve, right? And as a result, you have what's called return map, right? Starting from here, you come back. That's why it's called return map, and it's one-dimensional because your cross section basically, it's not one-dimensional cross section. It's where we maximized. It's two-dimensional, but again, it looks like a line. So when you come back right at the same position, that means you have a fixed point, right? You see, there's another fixed point over here, and then the other orbit that's shown here that starts here, right? It makes one revolution, lands here, then it makes another revolution, comes closer to the end, and then after that get back to this point, which means it's an orbit of period 3. Okay, and we, as I said, we don't touch the surface, right? We don't apply external current. Instead, we only shift the slow nucline up and down by changing that parameter. And the result, we're going to get what's called numerically, we're going to get a one parameter family of the one-dimensional maps, return maps, for the minimal voltage. And now you see how it looks like. So if I pick this value, for example, I have a stable fixed point here. So clearly, I'll converge to this attractor. And then that attractor, or the fixed point, will become unstable here. So I should expect period doubling, right? And then I'm going to expect something else. Or on the contrary, if I start here, then I should have a period doubling cascade. And that's what we're going to explore next. Okay. So we're going to begin with the upper map, this one, and then we're going to lower it down, so increasing this gap. As you can see, I said earlier that the shape of the one-dimensional map, okay, it's typical for square wave bursters, so no surprise, they all look alike, right? So let's discuss what we have. So we use this approach, right, and we found, so we're gonna know the surface, right, and then say computationally we find many points here, right, and then release them and then found the next image, and that's how the map looks like. Okay, don't get fooled, right? This is not analytical map, this is computational, but it's done so well, it creates an illusion that's analytical map. There is no way I can really feed this map, how it changes from this shape into that. I mean, I can, but it's not worth it. It's not the purpose. The purpose is to illustrate and demonstrate how it's done. Okay, and clearly this map has a single fixed point over here, right, which corresponds to a single point, which means it corresponds to a single local minima of this periodic orbit, and this periodic orbit is stable. So as you can see, this voltage in time, yeah, it's called tonic spiking orbit. Okay, then I I lower the nuclide. Okay, and then my map now has another attractor, which is orbit of period 2. So the old point here becomes unstable through the period doubling cascade and gives me orbit of period 2, which corresponds to orbit of period 2 on this manifold. Okay, and then something interesting happens, right? Then that orbit of period 2 becomes unstable, and now I have orbit of period 4. So look at this, I have 4 points, 1, 2, 3, 4, right? In addition to another periodic orbit, I didn't continue map long enough. Okay, and then I have period 8, right? And if you look at the papers that I just showed, then we have we demonstrated that you undergo a period doubling cascade as again that drop, right, gets steeper, which means the derivative becomes steeper, okay, greater than 1 in absolute value, right? So which means small negative 1 here and greater than 1 here. then you have contraction in the map, right? And again, you don't run away, which means the map and the floor that this map is generated with, right? They exhibit the same dynamics, right? So instead of studying the floor, you're welcome to study one-dimensional maps, and they basically tells you almost everything that you should know about the floor. Okay. On the contrary, if we're going to start with the other end of the map, this one, right? So let me see where we are. Okay, so that's how the map looks like. It's flat. This point is stable. Again, that fixed point corresponds to a single minima on the stable orbit. It corresponds to an spiking. Okay, then no surprise. Okay, when you transition from this state to that, you make this point unstable. It becomes a saddle, periodic orbit in the floor, and now you have orbit of period 2, which gives you double spikes or duplets of spikes like that, right? And then, okay, when you move this section of the map further, then to get to that tractor, iterates will need another step here, okay, which means you're going to get orbit of period 3, right? Which means orbit of period 1, orbit of period 2, orbit of period 3, next orbit of period 4, and then you have what's called period edging cascade. Okay, and that fixed point, by the way, it's corresponds to a saddle orbit. It's shown here. You can identify it, right, if you're good enough. And then these iterates, right, may approach this stable fixed point, and this is called a homoclinic orbit to this unstable fixed point. And as soon as you are in the vicinity of the homoclinic orbit, then your dynamics can become chaotic. So as you can see, this more or less, I mean, we don't show many iterates, but just a few to demonstrate that it's chaotic. And these are the flow, okay, these are these solutions of the neural model, and then they generate 3 spikes, 2, 4, and so forth and so on, right? So, which means the number of spike changes with every iterate, okay? And again, this is just a zoom in of the story, but if you wonder why this particular model, right, why it at the transition between 3 and 4 spikes becomes chaotic, the answer is that because the spike edging cascade occurs through what's called period homoclinic bifurcation. As soon as you approach the homoclinic bifurcation, as you can see right here, you kind of cycling around, right, diverging from the fixed point. Slope is very steep, which means you have strong instability. So that's basically the ingredients that you need to generate chaos in the system, right? Okay, and yeah, and that's what I showed you, right? But again, the building bricks, the building blocks of chaos in such systems are always homoclinic bifurcation. So, and this again, you see the orbit, okay, that starts at the critical point. It jumps right onto the unstable fixed point, which corresponds to a homoclinic bifurcation or something like that. And you can use the same, I mean, the tools of linear dynamics to figure out, I mean, again, to study homoclinic bifurcations or argue. Okay, and this time it's a long orbit, but still it comes close to the homoclinic orbit, right? And so forth and so on. Now, this is done, this is how you can derive the models if you're a skillful researcher, right? You can basically find these maps and they look almost like analytical maps. But if you're lazy, then you can try to extract the maps from the voltage data. I showed you before, right, how you do that. Okay, and you're gonna get, like, if you have orbit of period 5, then you're gonna get just 5 points like this. Okay, and you don't see how the branches are connected, but you add up a little bit of noise. That noise will kind of give you the variability, and then it's going to give you, it will try to fill in some missing gaps in a map, so you can kind of predict how the map is going to look like. Okay, now if I want to explain bistability in the system, right, I can use maps as well. But speaking of bistability, I showed you this before. So you can start as a tonic spike orbit or bursting orbit, and then by applying positive or negative pulse, you can switch between these orbits, right? So that means that's how you can explore bistability. But if you would like to find out how this map explains bistability, let's consider this particular map. Okay, so now look at this. I have this unstable fixed point, right, that corresponds to this red orbit, but also this part, after I change the parameter, then this quadratic tangency is going to give me two stable fixed points, right, like that, like the blue sky catastrophe. So one is going to be stable, okay, this one. The other one is unstable here. And then this unstable fixed point, this one, right, it bounds the attraction basin of this stable fixed point. So only trajectories that starts on the left, or on the left from this green, dark green orbit, or on the right from this red, will converge to this attractor, right? Otherwise, okay, you're going to get a bursting activity that has 7 spikes, if I'm not mistaken. So basically, that's how you use one-dimensional maps to explain biostability in a system. Okay, so speaking of the other bifurcations, we talked about the blue sky catastrophe, right? And this is another, was actually a original model when I found and analyzed the Lukyanov-Shilnikov bifurcation. Again, this elephant with the hobbit, right? So that's how it works. It's the same story. I showed you these pictures, right? So if I happens to be, again, this unstable manifold happens to be always on the right from the strongly stable manifold, then you have coexistence of bursting activity and tonic spiking. So if you happens to be only on the left, that eventually, or after one revolution, right, this bursting activity goes down and then converges, so limit cycle, unstable cell periodic orbit is right here. It's right here, actually, right? Then you converge to the stable tonic spiking activity. And in between, right here, you can generate, again, right here, you generate tonic spiking activity like this, and then some chaotic transition towards that tonic spiking activity. Okay, so where we are, and finally, so I click here, then let's say we go to Dropbox, the files that we share, higher School of Education, we click M files, click this 3D leech, and let me click at this file. If I run this, okay, then this is basically the activity that I can see here. Let me just pull it out. So the parameter, okay, in this model is this K2 shift. So you can see I have bursting activity. And let me make this parameter, say, slightly change it, right? Just a little bit. And guess what? You see, the model no longer burst, but instead it gives me a tonic spiking activity like this, right? If I go back to 2 again, then it becomes a burster. So clearly in between I have a transition. So if I make some number between these two values, right, then as you can see, bursting activity increased in size. Okay, so make it 7 and run it again. Oh, it gets even longer, as you can see that. Right, maybe we should integrate it longer. So where is the integration time? Let's make it 140, integrate it longer. Okay, like this. So no, maybe it's too much. Let's make it 8 here, so we can visually inspect how the burst duration increases. Okay, so one more time. How about 9? Because remember, 2, 3, I have tonic spiking activity. Okay, you see it gets longer, which means I'm approaching the blue sky catastrophe, right, around this value. So also is going to be part of your second part of your project, right? Then I click leech. Let me see, I don't remember what that. Again, remember the parameter here is this number, okay, and I run it. And this time, oops, let me just close the window and reopen it. Okay, now you can see, oh, wait a second. All right, so this time you can see this blue surface, right, which is sigmoidal curve. Okay, I'm pretty close to the knee point, and this is bursting activity, and basically I show you how you can detect these voltage minimas, right, these functions called events, right, and this allows you to find this red dot that corresponds to minimal voltage activity like this, okay. And let me see, the other file, it's called leech return map. Let's just run it, okay. Around this value, 225, run it. Okay, so, and then I added up some noise in the system, and basically this file is supposed to give you the shape of this one-dimensional map that we just discussed like this, right? But it doesn't look like, it doesn't look well pronounced over here, right? So I'll close this part, and then, okay, so the very last of the conversation, let's talk about what's called Fitzhugh-Nagumo plus Rinzel. John Rinzel, basically, this is a Fitzhugh-Nagumo system, right, which gives you a cubic null line, right, and then tonic spike, and then you add another equation, this one, and that equation basically changes, it makes the system three-dimensional, right, and that's the manifold, how it's going to look like. I mentioned that to you. So if you start here, you will be sucked into this region, right? And then this is the Andronov bifurcation. You lose stability, spiral away, end up on the outer surface, right, to generate the spiking activity. Then it transition towards the fold here, and then from that fold, you land back onto this dark blue line, right, to start a new cycle of bursting. And as you can see, in some setups, right, this model will generate elliptic activity, but its elliptic activity can be regular or it can be chaotic, right? And yeah, one more time illustration, if you make mu parameter smaller, again, in this case, slow null climb, this plane is given by this constant c, you shift it up and down like this, okay? And if I make mu smaller, that means I have more time to converge to the equilibrium state, right? Then I pass through the Andronov bifurcation, you see a spiral away, go up, but this time I go up basically this above this gray plane, right? So on average, happens to be travel always on the left, right? And when I'm below it, I always go to the right. And if I make mu smaller, then I generate activity like this, right? And get different projections. Okay, and the way the map is designed, in this particular case, one-dimensional map. So this is the movie, let me just run it. Again, this time I use a line on this manifold that corresponds to voltage maximus. And then you see I turn around and then find another point on this line, okay? As a result, this is basically illustration that I have more points, and then I populate this line with more points, and I'll create a map, one-dimensional map that looks like this. And then by changing the position of this low null line that's not shown here, right, I will get computationally one parameter family of the maps. If I just pick a few, then that crazy picture tells me that I have to transition from the upper shape, right, to that shape somehow. And transition is really crazy, right? So if the map is something like that, then I have a single stable fixed point that corresponds to tonic spiking activity. And then basically what happens here, this periodic orbit, I push it towards the fold, and that's where the crazy stuff starts occurring. Okay, so that map knows that this fixed point should lose stability very soon, and that map helps to achieve that, right? It gives a little drop here again, right? Then you have cell node bifurcation, and then you need this fixed point basically to merge with that stable fixed point and eliminate that, right? Eliminates that, and then you have what's known as the mixed mode oscillations, when you have a number of subthreshold oscillations, right? Like when I show you subthresholds like this, right? This is called mixed mode oscillations. You have subthreshold oscillations and then spikes, and those numbers change either chaotically or periodically, right? For example, 5-3 or I don't know, 5-7 or 3-4. So, and then you can create some kind of analytical stuff. So, and then you got something like this, okay, and then the map changes from that shape into that one, okay, and then that again this drop will lower in size, and then you have something like a unimodal map that you studied with Arkady Pikovsky, right? And then it has period doubling cascade reverse over here, and after that, okay, you're gonna get orbit of period 4, orbit of period 2, orbit of period 2, shrinks into fixed point, right? And then you have this period doubling cascade if you move backwards. Orbit of period 1, orbit of period 2, 4, 16, and then back to chaos, right? And that means you have basically, that means that in this case, right, you have chaotic subthreshold oscillations, and then after that you get large spikes. And this basically concludes our series of lectures. Okay, where is this thing? So I turned the camera on, and that's basically it, right? So I told you that using the limited time that we had, what you should know about mathematical neuroscience and how we use dynamical systems to approach modeling of individual neurons and neural networks. Of course, time is limited, right? Of course, I told you just the tip of the iceberg, but hopefully you found some aspects of this field interesting, and maybe you would like to pursue to move in this direction, right? And I mean, I have nothing special to encourage you about other than that you're always welcome. Google my name, right? I always use the same pathway. And then we've got some publications on this topic, right? So a lot of publications on the topic. And you're welcome to examine and review these papers. And tomorrow, I will have the rest of the day today getting ready for the lecture that I give tomorrow at 5 p.m. Of your time, right, at the Shilnikov workshop. So I need to get ready. And after I'm going to give a talk tomorrow, I'll shoot you an email saying what the second part of the project should be. And again, we will have no exam because there's no way I can verbally or orally talk to everyone, but you need to submit either PDF of your LaTeX project, right, in LaTeX form or as a PowerPoint presentation. Okay, at this note, I'm gonna end up stop talking, right? And if you still have questions, you're welcome to ask them right now. Thank you. Hello, no questions?
Ну, видимо, пока больше нет вопросов.
Матросов нет вопросов. Хорошо. Тогда я побежал, у меня начинается другой митинг.
Угу.
И всего вам доброго. Сегодня какой день? Четверг, да?
Да.
Ну ладно, спокойной ночи или там доброго вечера вам. Я помчался на другой митинг, ребят. Спасибо большое за внимание. Приятно было с вами пообщаться.
Спасибо, до свидания.
И я попрошу
До
Лешу не
свидания.